Why, hello everybody. It's still barely March 17th as I'm recording this. It's still Tuesday. I said I was going to work on the RNA SPRAP, like the genome application for the coronavirus. This is the one out of Wuhan. There's been work done. I'm getting excited. It's looking like something. Who's going to solve this $1 trillion problem? How are we going to defeat this virus? We're going to do it with the maths. We're going to do it at the molecular level. We're going to whoop this virus's butt. So here's what you're looking at. The virus is a bunch of nucleotides or bases long. Okay, nucleotides are the things in RNA and DNA. It's A, T, C, and G, and those are adenine, guanine, cytosine, thymine. The A's and the T's bond together, the C's and the G's bond together. Hopefully you're someone who knows something about genetics, and you know, I'm just telling you what you learned on day one of class. But if you're not, I'm going to try to make this video sort of for everybody. I've assembled the information from the genome of the virus in the order of people who are super smart and have annotated the genome already. They identify the genes or the strips of the DNA on there, and they call them certain things, and they put them in order. Well, I finally figured out how to get this whole thing into a table and put colors next to it so that we can look through it and try to figure out what the hell's going on. So I'll tell you my basic knowledge of what's going on so far. First of all, there's an order here. If you started at the 5' end, as they call it, the beginning of the genome, that part is 265 bases or nucleotides long. I have some other things called molar mass and density and hydrogen bonds that relate to each one of these nucleotides, and I add them up or I average them. I average the hydrogen bonds, I average the density to try to get a color, and here's why. We're just going to do the 5' end in this video because I don't know anything about genetics yet, and I'm going to learn what all these proteins are. There's these things called surface proteins and envelope proteins and nucleoplasmid. What is this thing? Nucleocapsid phosphoprotein. We got to learn what all those things are to figure out exactly how we're going to take advantage of them when we take out this virus. But I want to talk about the 5' end because I want to talk about the visualization of what's happening here and why this method of looking at the genome is something that I can pretty much guarantee that no geneticist has ever really had before, because nobody does this in Excel except for me. I'm the crazy person that does everything Excel. And so let's look at the 5' end right here. Okay. Now in the 5' end, now all we're just looking at is the genome from order 1 down to like 260 or whatever it was, right? And what I've done is I've opened up, this is the base pair right here. A stands for adenine, T thymine, right? C's. I color coded them. I made A's and T's green and yellow because they bond together. And I made C's and G's red to black because they bond together. So here's what I want to do, and kind of how we need to visualize things through numbers and eventually the 3-dimensional shapes that they make, but I want to try to go from 2-dimensional to 3-dimensional. So this is what it looks like in order as it's being coded. And I mean, you start to see how it's heavier in A's and T's at the beginning here. You get your first G at spot 7 or spot 8, and I'm not using the correct terminology for all these things. I'm making it up as I go along, so bear with me. Here we get hit with more C's and G's, which the C and G is a stronger bond. It takes 3 hydrogen power bonds or whatever to break it up instead of 2. So I guess the CG is a more powerful bond than the AT. The T is less dense than other things. The thymine's less dense. The C is pretty dense, but the G looks like it's the densest and it weighs the most. So that's the bigger one. So as we go down here, we have this weak area of average density and low density. I'm just looking at the green means big, red means small, yellow is in the middle, right? For these 4 things over here. And so what we're trying to get a handle on is, and then here starts to get choppy and get spotty. And this is just the beginning end of the genome. It's not even coding a protein. There's a bunch of A's here. We're going to do some other things to translate this stuff into more than just colors, but also somehow shapes or strengths or things like that. Does it really look like anything? A lot of A's and T's, strips of A's and T's, and then a C. Every nucleotide supposedly matters in the order of things. And right, I don't really know what we're looking at yet, but I know that there's going to be something we can do with it. So I'm running out of time, but that's what we've done today, and we're going to keep working on it tomorrow.